welcome again. Good afternoon. This is our newest installment of the Units of Life seminar series. As a reminder, given the fact that many of you may be new to this, and it's a relatively new seminar series, we started this with the idea that maybe we could have a place where we can convene to discuss deeper mechanistic biology. Here at the Broad, we're really great at using a lot of omics technologies and really doing amazing work with that. But sometimes it's really interesting also to see how we can go deeper into some of these biological mechanisms and learn more. And so we thought that maybe cross-cutting across all areas, we could have a seminar series that's more dedicated to that. So as with many things around here, this is also an experiment, which I sort of took on, and Jillian Shaw has been my partner in sort of organizing this. And so this is really the second year that we're running the seminar series, approximately once a month, sometimes every couple of months. And as you can see from the slides that are flashing through, we have some really amazing speakers, including Paul Cohen today, and some incredible speakers coming up. Joan Brugge, Helen Hobbs, Ardem Potapushan is coming for a special seminar in combination with MIT and Harvard. And it's going to be on a Tuesday for that particular time. And then Anna Maria Cuervo and Arlene Sharp to close out the year, and really amazing speakers lined up for next year as well, I promise you. And so without further ado, I wanted to actually introduce today's speaker. Paul Cohen is the Albert Resnick Associate Professor at the Rockefeller University. He is actually the director of the head of the Laboratory of Molecular Metabolism there. And he's, however, no stranger to Boston. As I learned, actually, he grew up not so far from here, but he also received his AD from Harvard College and then went to New York to attend the tri-institutional MD-PhD program, which includes the Rockefeller. He did his PhD with Jeff Friedman there, and then went on to Columbia for his residency training, and then came back to Boston to the Brigham and the Farber for fellowship training and postdoctoral work, and then back to New York to join the faculty at the Rockefeller, where, as I mentioned, he now heads the Laboratory for Molecular Metabolism. Paul has actually done some really interesting and remarkable work in adipocyte biology and really understanding adipocytes from the perspective of both his cardiology training, but also links to cancer, which makes him a very, it's a very interesting and unusual combination of trying to understand, the intercellular crosstalk and how this works with different comorbidities related to obesity. And so I think that we're in for a treat. So Paul, thank you so much for joining us, and we look forward to your seminar. Oh, I should mention before I go that there is a Q&A for those online, and Jillian has kindly agreed to moderate that. So we will be monitoring that for questions. And of course, everyone in the room, please feel free to ask questions. Because we're doing this in a hybrid form, we have mics, otherwise our friends online would not be able to hear. So try to make an effort to go to the mic, and we'll go from there. Thank you so much.
It's been a great day visiting with old friends and some colleagues I hadn't met before, but his work I've followed and admired for a long time. And as Anna mentioned, I was in this area for nearly 8 years and actually lived just a few blocks away. So it's kind of neat to walk around my old neighborhood. At any rate, please feel free to interrupt me if you have any questions. There should also be plenty of time for discussion at the end, and I'll try to point to things on the screen here as well. So for me, the backdrop to our work is this huge biomedical problem of obesity. And as you know, this is a problem that the United States is particularly good at. And by 2030, it's estimated that 50% of adults in the United States is going to be obese. But this is actually no longer just a problem of industrialized nations. It's a global problem as well. And for the first time in our history as humans, diseases due to overnutrition are actually more prevalent than diseases due to undernutrition. And so, of course, obesity per se, or the weight on the scale, is not the issue. It's all of these comorbid diseases that come with obesity: heart disease, diabetes, kidney disease, many different kinds of cancer, And over the past few years, as we've unfortunately learned, worse outcomes with COVID-19. And just to kind of highlight at a more personal level what got me interested in this question, I now see patients at Sloan Kettering, which is a cancer center, with a focus on patients with obesity and cardiometabolic disease and cancer. And it's surprising how often we see people with phenotypes like the two here. The first is a 75-year-old man with obesity, hypertension, hyperlipidemia, coronary artery disease, atrial fibrillation, and congestive heart failure with rectal cancer. And the second is a 52-year-old woman with an array of comorbidities, including obesity, coronary disease, congestive heart failure. She's had lymphoma, breast cancer, and newly diagnosed lung cancer. And I don't mean to imply that their health problems are solely due to their BMI, but clearly it's a major contributor to their pathology. And so as we think about these comorbid conditions, the fundamental question of my group has been to understand how this occurs. And we feel that the answers can be found, at least in part, by studying the biology of fat or adipose tissue. And so our work focuses in two areas. One is the biology of white fat. These are electron micrographs of white or brown adipose tissue, and we'll discuss each of these a little bit more. And so we are interested in what mechanisms link obesity to these diseases, and in particular, how does white fat contribute to obesity-associated disease? And then secondly, and this will be my focus of today's talk, might brown fat actually be an answer or a way to unlink obesity from these diseases? And so we've been asking, can it protect from obesity-associated diseases? And if so, how would that work? And so our approaches really are more focused on a question than a particular methodology. And so as a result, they span from cell biology to tissue homeostasis, into organ crosstalk, and translation into humans. And I'll touch on each of these in my talk today. So just to highlight a little bit more about the cell biology of fat cells, white fat cells have evolved to store excess calories in the form of lipid. They have these large unilocular lipid droplets which store energy as triglyceride. Very few mitochondria, and when energy sources are scarce, the lipid can be mobilized by lipolysis to provide free fatty acids as a source of fuel. Brown fat cells, on the other hand, have these small multilocular lipid droplets. They have a very high number of mitochondria. In fact, it's the iron in the mitochondria that literally gives tissues containing brown adipocytes a brown color. And rather than storing energy, these cells can actually dissipate energy as heat by a process called uncoupled thermogenesis. So this just illustrates how that process works. You probably remember from your biochemistry class days that in oxidative metabolism, what happens is that reducing equivalents enter the mitochondria and are passed along the electron transport chain. A consequence of this is the generation of a proton gradient across the mitochondrial membrane. And normally that proton gradient is used to power the synthesis of ATP through complex 5 or ATP synthase. And what is unique about brown adipocytes is that they express a protein called UCP1 or uncoupling protein 1, which sits in the inner mitochondrial membrane and is a proton fatty acid symporter. And so what this protein does is it allows the protons to leak back across the inner membrane, thereby dissipating the gradient and creating a futile cycle. Because the gradient is lost, all of the upstream steps are upregulated to try and reestablish this gradient, which of course can never happen. And because many of those reactions are exothermic, the end byproduct of that is the generation of heat. And we know that these cells not only can generate heat, but from mouse studies dating back many years ago from Brad Lowell and others, if you ablate all UCP1 positive cells in an animal, this results in a phenotype of cold intolerance and obesity. And it makes sense that these cells have a role in metabolism because in order to do this, they need fuel, and they help clear a number of toxic metabolites from the blood, including glucose, free fatty acids, and branched chain amino acids. So in terms of its evolutionary role, we think that we developed brown fat to protect against hypothermia. And for mammals, brown fat is a mammalian adaptation. Hypothermia is a major threat to early life because we're born small and hairless with a fairly large surface area relative to our mass. And so here I'm just showing you, Oh, that's not on my screen, so I don't have to do anything. Here I'm just showing you thermal camera imaging of a litter of newborn mouse pups. And what you can see, these are some newborns all lying in a pile on top of one another. This pup has gone venturing away from its brethren, and you can see this very strong heat-emitting signal on its back between its shoulders, and this is the so-called developmentally preformed intrascapular brown fat. And this same depot of brown fat is present in newborn humans in an interscapular location. We also know, though, that mammals have inducible brown fat. So in response to cold, fat has the ability to turn on a program of thermogenesis in a type of cell called beige fat cells. And what I'm showing you here are dissections of mice where all you see is the fat and the associated organs. And this is from a mouse housed at 28 degrees Celsius. You can see the developmentally preformed brown fat. But after housing the animal at 6 degrees Celsius, where it can happily live for an indefinite period of time, you can see the clear change in the appearance of the fat. And this represents the induction of these multilocular beige adipocytes with a high number of mitochondria, giving the tissues the brown color. And so I mentioned there is this second type of inducible brown adipocyte called a beige adipocyte. These cells have many of the same properties as brown adipocytes, with one of the main distinctions being that their activity is highly inducible. So in response to cold, beta-adrenergic agonists, exercise, or thiazolidine-dione drugs, these cells can increase their thermogenic activity. And importantly, we know that the brown fat that we can detect in humans shares a transcriptional signature that's very similar to rodent beige fat. And so we think these are a particularly useful model system for studying thermogenic fat. At a transcriptional level, the identity of these cells is dictated by a transcriptional co-regulatory protein called PRDM16. And in early work in Bruce Spiegelman's lab, where I was a postdoc done by Patrick Seal and Shingo Kajimura, they showed that PRDM16 is required for the formation of thermogenic fat cells. So if you ectopically express PRDM16 in a fat cell, it gives it the molecular, morphologic, and bioenergetic properties of a thermogenic fat cell. And the way that PRDM16 works in fat cells is not by binding DNA itself, but by contacting other transcriptional components, including PPAR gamma, PGC1 alpha, and the mediator complex, to activate the expression of brown and beige fat selective genes. And then it can also bind to CTBP and histone modifying enzymes to modify the chromatin to suppress white fat genes. So it works in two ways to dictate the development and maintenance of these cells. And so in work that I did as a postdoc, I generated and characterized a mouse model that has an adipocyte-specific knockout of PRDM16. And because this knockout is done postnatally, the mice specifically lose their inducible beige fat, but not their preformed brown fat. And so as a result, they do not have any difficulty tolerating the cold, but they do have many features akin to metabolic syndrome in humans, including more visceral fat, mild obesity, insulin resistance, and hepatic steatosis. And so this finding and many of the others that I mentioned, as well as a whole body of work that I don't have time to discuss today, has led to this great interest in the field as to whether we could perhaps target thermogenic fat for therapeutic benefit, either as an anti-obesity agent or as a way to mitigate against obesity-associated diseases. However, until about a decade ago, I think most people would have told you that was a waste of time. And the reason for that was because the feeling was that thermogenic fat was really important in small mammals like mice and in newborn humans, but that in adults, brown fat atrophied and was no longer relevant. And that notion really was overturned with a series of papers published in the New England Journal of Medicine in 2009, showing that adult humans do in fact have cold-inducible brown fat. So what I'm showing you here is an FDG PET CT scan. These scans measure the uptake of radio-labeled glucose. And then the CT scan can allow you to see the imaging characteristics of the tissues that take up the glucose. And in this particular study, young healthy subjects were imaged either at thermoneutrality, which is how most of us live, or after just a couple of hours of moderate cold exposure. And you can now see the appearance of these FDG avid tissues along the neck and cervical spine. And in one of these tissues, subjects actually allowed themselves to have biopsies taken of these tissues, which is daring because these are close to some vital vessels. But the tissue obtained showed that these cells here have the morphologic and molecular characteristics of brown fat. And so most of our work since then has come in two forms, the field's work, I should say. So either retrospective studies looking at series of PET scans and linking them to clinical data, usually numbering up to a few hundred patients, or small prospective studies in which roughly 10 to 20 subjects, typically healthy young male subjects, are prospectively exposed to cold, and then some metabolic readout is studied. And so these studies have been incredibly useful in suggesting that brown fat promotes insulin sensitization and glucose lowering, but because of their size, they weren't really powered to look more comprehensively at the health effects associated with brown fat. And so that question motivated Tobias Becher, a former fellow in my lab, who was interested in this broad question of, does brown fat protect against obesity-related comorbidities? And to do that, he took advantage of clinical PET scans. So unlike the research scans I showed you where subjects are deliberately exposed to cold to try and bring out brown fat, in clinical scans, if anything, patients are kept warm to suppress brown fat activity because these scans are done to diagnose or track the progression of cancer, and you don't want any extra signal. And what is clear if you start looking at these scans is that there's a tremendous natural variation in brown fat. So you see this individual has quite a lot of brown fat, whereas this individual seems to have hardly any or perhaps none at all. And so Tobias and I quickly realized that because I had a clinical role at Sloan Kettering where they do somewhere between 10 and 20,000 PET scans a year, this would be a rich resource for looking at associations between brown fat and health outcomes in humans. And so what he did was he retrospectively reviewed about a decade worth of studies, and because it's their habit there to always comment in their reports as to whether or not brown fat is seen or not, we could look through all of the reports and segregate people into either brown fat positive or brown fat negative, and then we could associate their brown fat status with all of the other information in their electronic health records. And so in total, this gave us a cohort of more than 50,000 patients with over 130,000 scans. The discrepancy is because a number of patients have multiple scans. So this was at least an order of magnitude greater in size than any series we were aware of. And so the other thing that we did then was to try and develop a matched cohort. So of course, an important consideration when one is doing these kinds of retrospective studies is you need to consider different covariates. And it was already well established that brown fat associates with age, sex, BMI, and outdoor temperature, meaning brown fat is less prevalent as we get older, unfortunately, less prevalent in males, less prevalent with higher BMI, and more prevalent in colder outdoor temperature. And so what we did was we took people with brown fat and matched them roughly 1 to 2 based on these 4 covariates to give us a matched cohort of about 5,000 people with brown fat and about 10,000 people without brown fat. And when we then linked that to the diagnosis codes in their electronic health records, we saw that the odds of a variety of cardiometabolic diseases were significantly reduced in people with brown fat. So most strikingly, type 2 diabetes, which had been described before, but also a number of associations that had not been described before, such as coronary artery disease, congestive heart failure, and hypertension. And importantly, because this is a matched cohort, these effects are obesity independent. So this has nothing to do with body weight or BMI. This is based on brown fat status. And if we look now by segregating people by BMI, and these are the different diagnoses, but I'll just draw your attention to type 2 diabetes here. If we group people into categories based on BMI, either normal, less than 25, overweight, 25 to 30, or obese, greater than 30, and then look at people with brown fat in brown and without in blue, what you can see is that for each BMI strata, people with brown fat have a significantly reduced odds of the pathology involved. And interestingly, if you look at people who are obese with brown fat, their prevalence of type 2 diabetes is similar to people who are overweight or normal weight without brown fat. We also can link brown fat status to laboratory values, and I'll just show you a couple here. So people with brown fat, again, matched for all of these covariates, have significantly lower fasting blood glucose. When you plot blood glucose as a function of BMI, you can see that without brown fat, there's a fairly linear relationship, and that is flattened or blunted in those with brown fat. And we see a similar relationship for triglycerides. So what Tobias next did in a follow-up paper was to try and better understand how. And so to do that, he got a little bit more granular with these scans. And because of that granularity, which required a lot of time actually manually reading scans, we only were able to do this on a subset of scans done over 1 calendar year. So this is a few thousand scans. And what he did in collaboration with Andreas Widmer was to first look at the CT scan. And by looking at slices from the CT scan, we could quantify the amount of either subcutaneous or visceral white fat. And the reason why we did that is because visceral white fat is particularly pathogenic, whereas subcutaneous white fat is comparatively less deleterious. And we wondered whether people with brown fat have a different type of white fat distribution. And then he also went beyond just this binary categorization of yes or no brown fat to actually quantify brown fat to see if we could find a quantitative relationship between brown fat and some of these other variables. And I just want to show you 2 pieces of data from this study. So first of all, we see that people with brown fat have a healthier white fat distribution. And what I mean by that is if you control for BMI, people with brown fat have relatively less visceral fat and more subcutaneous fat. And also now, if you do a regression analysis where you look at the amount of visceral fat as a function of brown fat activity, you can see that people with increasing brown fat activity have successively lower amounts of visceral white fat. We also looked at the CT scans to try and assess liver fat. So in prior work, it's been shown that the liver density as measured by Hounsfield units is correlated with liver lipid storage, such that higher liver density is associated with lower liver fat. And we saw, again, matched for all of the covariates, that people with brown fat have a higher liver density, meaning lower ectopic lipid storage in the liver. So in the next, I guess, 20 or so minutes remaining, I want to talk about our work, which has really been focused in understanding what is responsible for these benefits associated with brown fat. And if we come back to our approach, I'll tell you about 3 stories that have to do with form. So first of all, how does thermogenic fat work? Secondly, function. How does it communicate with other tissues? And third, regulation. What are the genetic determinants of thermogenic fat quantity and activity? So in terms of the form, this is work that was led by Jingyi Qi, a former PhD student who's now a postdoc in Boston, as well as Rico, a current graduate student, and Will Barr, who's actually in the audience and is now a graduate student at MIT. And they were interested in understanding what are the cellular and molecular components that control the formation and function of thermogenic fat. So just to give you a little bit more introduction, because we're doing these studies now in mouse models, in mice, there are 3 main fat depots. There's the developmentally preformed interscapular brown fat that consists largely of brown adipocytes, as I mentioned earlier. These cells have a high number of mitochondria and dissipate energy through thermogenesis. Visceral fat is largely white adipocytes and stores energy, but subcutaneous fat, which was the focus of our studies, contains a mixture of white adipocytes and beige adipocytes that in response to cold can turn on this thermogenic program and dissipate energy. And so at the onset, we already knew quite a bit about beige fat cells, mainly through studies either in vitro or in animal models. So first of all, activation most classically occurs through cold stimulation through the sympathetic nervous system. When these cells are activated, they're marked by the expression of this protein UCP1, which is crucial for uncoupled respiration. And this is dependent on PRDM16. And when you activate these cells, this is associated with metabolic benefits, including reduced body weight, improved insulin sensitivity, and improved glucose homeostasis. At the beginning of our studies, though, there was also a lot that we did not know. And in particular, we didn't have any sense at the level of tissue, how do these beta-adipocytes interact with other tissue components? And one of the main limitations, which is highlighted here, has to do with how do we look at how cells relate with one another in a tissue. And so in traditional approaches, that's done histologically in these two-dimensional sections, either with paraffin-embedded sections or with immunofluorescence. And that presents a particular challenge because of its high lipid content, which makes it challenging to obtain consistent sections. And then the lipid can also cause light scatter. And so what Jingyi did, working in collaboration with people from Marc Tessier-Levine's lab, is to develop a protocol that we call AdipoClear, which allows us to visualize three-dimensional structures in adipose tissues with molecular resolution. So the starting point is that tissues are cleared or rendered transparent to light. This is a fat pad before and after clearing. And then they can be visualized after staining with antibodies by light sheet fluorescence microscopy. And I'll talk to you about some of her work, but this has also been really useful in a number of collaborations that I highlight here. And so to start, we asked where are beta-adipocytes located. And so this is the subcutaneous fat pad in a mouse. And we, for naming convention, we call this portion the inguinal portion. There's a large lymph node here. And then on the other side of the lymph node is the dorsolateral portion. And generally in the past, people didn't consider regionality of the tissue. So if they looked at the activation of these cells, they would take an animal, expose it to cold for varying lengths of time, and then either measure the level of transcripts or proteins involved in the process. And you can see at both the mRNA and protein level, there's a really robust upregulation of UCP1 with cold exposure. But when we did whole tissue imaging, and I'm showing you now UCP1 in magenta after 48 hours or 1 week of cold, you can see that there's pretty striking regionality. So after 48 hours, most of the UCP1 positive cells are in the inguinal portion of the tissue. And then after 1 week, more cells emerge here. They also begin to emerge in this aspect of the dorsolumbar portion of the tissue. So why is that? Does it have to do with the environment where these cells live, or are there intrinsic differences in the fat cells in these two regions? And we actually think that both may be true, but for today, I'll focus on the environment and in particular, the role of the sympathetic nervous system. So the way that the sympathetic nervous system activates these cells is highlighted here. So cold is sensed in the periphery by sensory neurons. That signal is conveyed to the brain. That then leads to an efferent output through the sympathetic nervous system, first in preganglionic neurons, and then in postganglionic neurons, which have their cell bodies in ganglia along the spinal cord, some distance away from the target cell. They send these long-range axonal projections into the tissue parenchyma, where they release catecholamines that binds to beta-adrenergic receptors in fat cells. That leads to increased cyclic AMP and a signal transduction cascade, the end result of which is the upregulation of this thermogenic program. And so here now I'm showing you imaging staining for TH or tyrosine hydroxylase, a marker of sympathetic neurons. And what you can see is this really rich network of pH positive neurons. You can see these large fibers traveling along blood vessels where they regulate vasoactivity as well as axons. But if you zoom in, you can see these finer puncta that enter the tissue parenchyma. And if you really zoom in, you can see that these are actually in close apposition to fat cells. And so we can also take these cleared tissues and do confocal imaging to get better images of thicker sections. And when we do that, what you can see is this network of parenchymal nerves. And if you look, you can see what looks like this beads on a string pattern or these varicosities. And these actually represent sites of neurotransmitter aggregation where they're then released en masse, and so that the cells closest to them get the highest concentration of catecholamines. And when we do this, we can also trace these projections to actually quantify their density. And so when we use these tools and we now look at the different regions of the tissue, Region 1 is this region that has beige fat cells. Region 3 has fewer. And if you look in these fly-through images, you can see that there's much greater density of pH in green in the inguinal region than in the dorsolumbar region. And when we quantify that by tracing these projections, you can see that there's about a 2 to 3-fold increase in neurite density in the inguinal portion of the tissue. So we next asked, well, how are these two processes related? And so to address that, what we did was we looked at either control animals or animals lacking beige fat cells because they have an adipocyte-specific knockout of this key regulator PRDM16. And what we saw was quite striking. So if you look in control animals, these are just single cut images. In the control animals, there's very dense pH staining in this inguinal region and very scant staining in the dorsolumbar region. But in mice lacking beige fat cells, there's very scant nerve density in both regions of the tissue. And that was really unexpected because what that tells us is that simply by deleting a key transcriptional regulator in fat cells, we not only affect the phenotype of the fat cells, but we somehow affect their innervation. And so this just summarizes our working model from this work. So it was well known that sympathetic nerves activate beige adipocytes, but what we found is that these beige adipocytes are actually required to reinforce their innervation. And if you delete PRDM16, you not only lose beige adipocytes, but you also lose this innervation. So that, of course, leads to this chicken and egg question of what's causing what. And in work I don't have time to get into today, we focused on whether or not this pattern of innervation is developmentally determined, or does it show some plasticity in adulthood? And if it does show plasticity, how is it affected by things like obesity, cold exposure, and aging? So in a follow-up paper, Jingyu was able to show that there is this critical early developmental period when the patterning of these sympathetic neurons is established. And at least in our paradigm, where we use inducible deletion of PRDM16, in adulthood, this interaction between beige adipocytes and the nerves is not required to maintain the structural maintenance. And so again, this suggests that this is regulated by PRDM16 dependent factors in the fat cell, and we're now intensely focused on understanding how that works. And so 3 possible modes of action. It might be that thermogenic fat cells release factors that attract nerves. Conversely, white fat cells may release factors that repel nerves, or there could be indirect interactions between fat cells, immune cells in the tissue microenvironment, and the nerves that dictate their pattern of innervation. And so we've profiled different regions, both by transcriptomics. We hope soon to do proteomic profiling, and we've identified a number of candidate factors involved in axon guidance, and we are now doing functional analyses of these factors. So let me now turn away from form and towards function and understanding how thermogenic fat communicates with other tissues. This has been really a team effort. It was initiated by Chan-Hee Choi, a former MD PhD student who's now finishing medical school. You can see Will Barr again features prominently here. And some of the newer work is being led by Kaya, a postdoc, along with Ori and Samir. And so you might ask to start, well, why should I even think that thermogenic fat signals to other tissues? What is the rationale for saying it has an endocrine role? There are a variety of studies, none of which prove this, but in my mind, this is the most convincing study. This comes from Dave Piston's lab at WashU in St. Louis. What they did here was they took a mouse model of type 1 diabetes. So these mice were treated with streptozocin to ablate their pancreatic islets, leaving them with diabetes with blood glucose values above 400. They then transplanted just a small amount of brown fat, about 50 milligrams of tissue. One would think far too little to consume all of that excess glucose. And when they did that, they saw this very durable correction of the type 1 diabetic phenotype that lasted out to 6 months. And this remains in the absence of insulin. So these animals still do not produce insulin, but somehow the transplanted brown fat is sufficient to nearly normalize their blood glucose. And so It turns out actually that fat cells, if you look bioinformatically, are predicted to secrete more than 1,000 unique polypeptides and probably an even greater number of small molecule metabolites and peptides. And the vast majority of these molecules, as well as their targets and functions, have not been characterized. And one of the major stumbling blocks has been a lack of suitable technologies, especially in the proteomic space. And of course, if we can solve this problem, we have the potential to answer big questions, such as the links between obesity and disease, how brown fat might protect and perhaps even lead to new therapeutic targets. And so for proteomic work, the major challenge, of course, is that proteins in the blood exist over more than 10 orders of magnitude in concentration. And so if one does traditional mass spec of plasma, you will detect a lot of albumin and globulin, but it's very difficult to identify proteins in the nanogram or picogram per mil range, which is where we think these hormones exist. And so we've used a variety of chemical approaches. I'll talk about one today called BondCAD, or bioorthogonal non-canonical amino acid tagging. Our initial studies, which I'll tell you about, were done in vitro, but we're now doing this in vivo, and I'll explain how that works as well. So the way that this approach works, of course, normally in protein synthesis, methionine, for example, is incorporated into the proteome by a methionine tRNA synthase. What you can do is replace methionine with a methionine analog, in this case called azidohomoalanine, or AHA. And this analog contains an azide group, which does not exist in amino acids in nature. And why this is useful is because from this point on, all newly synthesized proteins will be azide labeled. And the reason why we care about that is because we can now do an experiment like this one, where we can take primary fat cells from either the brown fat depot, subcutaneous depot, or visceral fat depot, replace the media with a media containing AHA instead of methionine, keeping serum there. And so all of the newly synthesized proteins will be azide labeled. We can collect those from the conditioned media, and then use click chemistry, where we take a bead coupled to an alkyne moiety, which will form a covalent bond with this azide group, use the bead to pull these proteins out of the mixture, and then digest them and do mass spec. And so that's exactly what we did. And when we did that with these three different kinds of adipocytes, we identified more than 600 unique proteins secreted into the media, and about 350 of them showed enrichment in the secretome of different kinds of adipocytes, which you can see in this heat map here. So here is a collection of proteins that are secreted particularly by brown fat cells. Here are proteins secreted by visceral white fat cells. And if you look at this functional characterization, you can see, interestingly, given what I was telling you in the previous story, that the proteins secreted by brown fat cells are enriched for molecules involved in axon guidance and neuron projection guidance when we do GO analysis. And so we, of course, don't just want to assemble lists of proteins, but we want to identify proteins and do biology on these proteins. And so Chan He's work, which was actually accepted for publication yesterday, focuses on a white fat derived protein that he shows can promote insulin sensitization. But we've also identified a number of other interesting candidates, including a cold-induced secreted protein made by brown and beige adipocytes that's almost completely unstudied, and the function of which continues to elude and vex us. So I hope to solve this problem one day in the not too distant future. We're also moving towards doing this in vivo, and the reason for that, I think, is obvious, but it's not possible to adequately model a variety of pathophysiologic conditions in vitro, like obesity, fasting, and cold exposure. And then secondly, as good as these cellular systems are, they are heterogeneous and somewhat artificial. And lastly, we don't want to just identify proteins that can be secreted by a cell, but proteins that actually circulate in the blood at physiologically meaningful levels. And so it's now possible to do that with a variant of the approach that I told you, where now a different methionine analog is used called azidonorleucine, or ANL. You'll notice, for those of you who are chemically inclined, that this has a more bulky side chain group. And so as a result, this is normally not well incorporated into the nascent proteome by wild-type methionine tRNA synthase. However, in this work done by Aaron Schumann's lab, what they showed is that making a single point mutation in the methionine tRNA synthase, an L to a G, now allows it to incorporate ANL into the nascent proteome. That is very useful because they have made a conditional mouse model, which they were kind enough to share with us, that has knocked into the ROSA 26 locus a lock-stop locks in front of this mutant methionine tRNA synthase. This allows you to then cross it to your pre-line of interest. And then when you introduce ANL coupled to whatever physiological state you're interested in, only proteins made in cells expressing pre-recombinase will be labeled. And so we can again now use this approach to enrich these proteins from the blood. And we believe that this enrichment approach offers us the potential to identify low abundance proteins that would typically elude us with conventional mass spec. And in principle, this chemical handle could even allow us to track proteins to their destination. So this just shows you a proof of concept of this approach. So if we use this mouse strain crossed to UCP1 Cree so that we label only UCP1 positive cells and house mice either at 30 degrees or at 8 degrees, you can see the Cree negative mice give almost no signal, so very low background. And we can see very clear labeling of proteins in the Cree positive animals at 30 degrees and 8 degrees. And you'll have to take my word for it, but if you look closely, there is some unique banding pattern of these two different temperatures, and we're now beginning to try and uncover what these proteins are that are actually secreted. We're also taking a variety of broader approaches. So I mentioned BONCAT. We're also using another chemical approach in collaboration with Alice Ting, Katya Vinogradova, and others using proximity labeling. This is also a Cree-dependent approach in which proteins in the ER pathway are biotinylated in a cell type of interest. And again, we can use this biotin handle to enrich the proteins in the blood. We're also doing bulk analyses of serum and plasma, both looking for proteins as well as lipids and metabolites. And then we've also developed an increasing program doing studies in humans. And I want to take a few minutes to tell you about that. So because this all originated in humans, I feel it's particularly important to study this problem in human subjects. And so we've developed two studies. The first is well underway. We call this the cold vest study, and this looks at changes in plasma proteins, metabolites, and lipids in response to acute cold exposure. And the second, which just received IRB approval and we hope to start soon, looks at the effects of chronic cold exposure. Let me tell you a little bit more about this. For the cold vest study, we recruited men and women, normal BMI between 18 and 28 years of age. We picked these conditions because we specifically wanted to look at people who had a very high likelihood of having cold-induced brown fat. Because of logistical challenges and cost, it's not possible to screen each of these people with a PET scan pre and post. But we excluded people with known pathology, any prescription medications, illicit drugs that might contribute. And so what we did was we recruited these people. They came fasted. We did some phenotyping and measured body composition. We then took a baseline blood sample and then exposed people to cold for a 3-hour period using a cooling vest protocol. This is a well-established method that was developed by Aaron Saitis and others at the NIH, where you put on it almost looks like a life jacket that circulates cold water. And the idea is that you customize this to each individual. I think we all know that different people have different cold tolerances. And so we progressively lower the temperature until the subject begins shivering. We call that the shivering threshold. And then we raise the temperature 2 degrees. And that's where the patient sits for the remainder of the study. And then at the 3-hour mark, we collect blood again. So we've done these paired studies on 20 subjects, and we've now done a variety of assays in collaboration with Rod Gersten and Alan Sagatilian, including SomaScan and Olink to look at proteins, metabolomics and lipidomics of the plasma, with the goal of identifying factors that are regulated by acute cold exposure, possibly one day leading us towards biomarkers, and also these might be functionally important candidates that we can assess further in cellular and animal models. So I just want to show you a bit of data from this work. This is all actively underway. This is from the metabolomic analysis, and there's about 400 or so metabolites shown here in this volcano plot. In blue are things that are upregulated in the cold, red are things that are downregulated. And I just highlight two as a proof of concept. So dihome is a lipid mediator that's been studied by Yuat Sang's lab at the Goslin and shown to be induced in cold in both animals and humans, and to also facilitate fatty acid uptake by the skeletal muscle. And what you can see in our subjects is this is regulated in response to cold exposure. We also see DMGV. Don't ask me to tell you what this stands for because I'm drawing a blank, but this was previously identified by Rob Gersten's lab as being a marker of hepatic steatosis and risk for development of diabetes. And you can see that the levels of DMGV go down in the blood with cold exposure. These are two of many interesting candidates that we're increasingly uncovering. The second study that we hope to soon kick off is called the MUI study, or Minneapolis Ice Water Immersion Cohort. And you may find this a bit crazy. This actually started by a group in Minneapolis contacting us to ask if they could participate in research on brown fat. And so there's actually a group of several hundred individuals who voluntarily cut holes in the ice and swim in these frozen lakes during these frigid winter months. And they feel that it provides them with tremendous benefits, both physical and psychological. And so we thought, well, maybe there's actually something interesting here. And so what we've done is develop this study where we will study these individuals both in the winter when they're taking these cold swims. Some of them do it every day. And then in the summer when they're not. And so for all of the individuals, we will profile their plasma proteome, metabolome, and lipidome. And for a subset of subjects, we'll also do deeper phenotyping, including hyperinsulinemic euglycemic clamp studies, FDG PET scan studies, and we'll also obtain fat biopsies to do single nuclear sequencing to look at the cellular and molecular landscape of their adipose tissue. So this was actually a long slog to get approval for this because it involves two institutions, since we're not where these patients are. And so we're excited to be collaborating with Betsy Sequist at the University of Minnesota and hope to kick this off very soon. So in the last, I think I have just a couple minutes, I want to turn to the regulation. So ultimately, I think if we're going to get anything actionable here, it's important that we understand what regulates brown fat in humans. And looking again at this variation in brown fat quantity and activity, it begs the question, what are the genetic determinants of this? That hasn't really been studied before. We know that obesity is a highly genetic phenotype. Might brown fat be under genetic inheritance as well? And so I just want to show you data from two other studies that at least suggest this isn't a crazy thing to think about. So this is from a retrospective study from Aaron Cyphus and Ron Kahn, where they looked at brown fat quantity in men and women. You can see women have more brown fat than men, as I told you. But if you look at the scatter plot, what you see is there are a small number of individuals who have exceptionally large amounts of brown fat. So in other cases, these extreme or exceptional variants can sometimes be used to identify single alleles with large effect size that contribute to the phenotype. This is from a separate study done in the Netherlands, where people did PET scans to measure brown fat volume of either Caucasian subjects or South Asian subjects living in the Netherlands. So it was meant to control for environment. And what you can see is that the South Asian subjects have a significant reduction in brown fat volume, at least consistent with this being under some form of genetic inheritance. So we're now taking several different approaches, and I'd welcome any suggestion. I know there's a number of geneticists in the audience and perhaps on the Zoom call. I'll just briefly mention what we're doing. So the first is that we've gone to our Sloan Kettering cohort of about 50,000 patients. This was a retrospective study, but fortunately, a number of the patients in our study have had their exomes sequenced as parts of other studies. And so we were able to piggyback on that. And we've specifically focused on people who either have exceptionally high brown fat activity greater than the 90th percentile for their age, or who have what we call age-retained brown fat. Since brown fat prevalence goes down with age, we reason that people over 50 who still have brown fat must be rather rare because the prevalence is like less than 1%. And so we have a cohort of about 100 people who fit those criteria, and we're comparing their exomes to reference exomes and have a number of candidates that we're beginning to study. We're also looking at two different well-defined cohorts. The first is the Turkish obesity study in collaboration with Tayfun Özelek at Bilkent University, where he has done exome sequencing on about 1,000 people with BMI greater than 40 from consanguineous pedigrees. And the hypothesis here is that these individuals might have mutations that would give them decreased brown fat activity, therefore decreased energy expenditure and predisposition to obesity. And then finally, the third cohort is in collaboration with Sadat Faruqi in the UK. She has a cohort of people who have a thin phenotype. This is not commonly done, but rather than sequencing people who are obese, she has sequenced the exomes of a few thousand people who are thin with a very low BMI, but have no underlying medical pathology that seems to explain it. And the prediction is that these people might have variants that give them high brown fat activity, increased energy expenditure, and protection from weight gain. And so the paradigm that we're following is to identify these variants, engineer them into cell lines, do phenotyping both at a molecular and bioenergetic level, and then for promising candidates, make animal models when the residues are conserved to see if the phenotypes match. And so just to summarize, with this backdrop of obesity and all of its attendant medical complications, we believe there's some reason for optimism in that brown fat seems associated with protection from cardiovascular and metabolic diseases. And so we've really focused now on understanding how that works. I showed you data about the form of thermogenic fat and how it can be regulated by crosstalk with the sympathetic nervous system. I've also told you about our work on the function, on the regulation, and the endocrine action of these tissues. And so the long-term goal, of course, is to use all of that information to try and develop therapeutics to break the link between obesity and associated diseases. And so in closing, I just want to thank all of the people in my lab, our collaborators, and I thank you all for your attention. I'm happy to take questions. I see there are questions in the chat. Do you want to?
I was going to say, Paul, the folks online have been very patient. So first, Ed Skolnick wants to know, is there a way to activate PRDM16 with small molecules and not require the sympathetic system to activate it?
Yeah, so great question. I mentioned some of the students who I had lunch with today. When I was a postdoc, I spent a few months doing a small molecule screen at the Broad looking for small molecule activators of PRDM16. That work, unfortunately, was a failure, not because the screen failed, but because we were too naive in our understanding of how PRDM16 was regulated. So at the time, we assumed it was regulated transcriptionally, and so we did an RNA-based screen, and we identified only 2 or 3 hits, none of which really bore out. It's now become clear over like the last decade that PRDM16 is largely regulated post-translationally. And in a recent paper in Nature from Shingo Kajimura, he identified the mechanisms that regulate the turnover of PRDM16. So I think those ubiquitin ligases that regulate the half-life and turnover of PRDM16 are probably especially appealing targets for ramping up thermogenic fat activity.
You can hear me? Yeah, great. Amazing talk. I really loved, and every time that I see your work, I get really super excited. I want to follow up this question of Ed, and also connect this with 2 recent works that is coming out from Lee E from Scripps, and also from Ken Law that was in George Friedman about the neuropathy. When you have obesity, but you have neuropathy, you lose sympathetic nervation in the fat, and linked together with this PRDM16 phenotype that you see that when you knock out, you have less innervation. So by what you guys see in terms of gene expression in the basal adipocytes through development, do you see loss of any neuropeptide or any growth factor that could be mediating innervation, or if you see even with the PRDM16 knockout, a loss of inhibition early on in development, and how obesity could play a role in that.
Yeah. Thank you for your question. What Hernandez was alluding to is work from others in the field showing an important role of sensory nerves, first of all, in regulating these cells. I focused on sympathetic nerves, and also on the role of leptin in regulating sympathetic nerves and how obesity may or may not alter that. So in our studies of the PRDM16 model, we used also a DOX-inducible system. I didn't have time to get into this. And we saw that if we delete PRDM16 in adipocytes in this crucial window early in life, then we lose the innervation, and that's not rescued later on. But if we delete PRDM16 in adipocytes in fully mature adult animals, we don't see any apparent effect on the innervation. So that has then led us to focus much more intensively on what's going on in this crucial early period. We've identified a number of candidates, and that focus on the early period has also led us into some interesting insights into where these beige adipocytes might actually come from. So I hope to have more on all of that soon.
So it's fantastic work. I was really curious about the VEST study, and you regulate by how much cold the individual feel. And is there a correlation with already having more brown fat or beige fat to feel more, and then this can be a variable in your study?
Yeah, that's a great point. One of the major weaknesses of our study, and I hope you don't review it one day because you've just highlighted it, is that we don't have PET scan data on our patients. So our research hospital is great for doing small studies. We don't have a PET scanner. Research PET scans cost a couple $1,000s. And so what we would really want to have is both a baseline and a cold-induced PET scan on all of the subjects in our study. We don't have that. However, we are going to be collaborating with Aaron Cypus, who's at the NIH, who has one of the greatest sets of such samples. And so the plan will be to look at blood from people with well-defined brown fat quantity and activity pre and post cold, and do the same assays both to validate our findings, but also to look at things that are maybe correlated with brown fat quantity and activity.
More virtual questions, or can I go ahead? Yeah, wonderful talk, wonderful work as always, Paul. So I have a few questions. I'm just going to be really quick, and I'm glad we have more time to discuss later. One is for the sympathetic nervous system interaction, which of course makes complete sense and has been modeled in the dish before. There are studies from Bruce Spiegelman and others showing just simply reducing the temperature of the incubator, inducing the phenotype of beiging in adipocytes. Could you comment a little bit on that?
Sure. Yeah, so that was work from the same Lydia that Hernandez alluded to when he was a graduate student. So this was a study where a former student took fat cells in a dish, so no nerve, no blood vessels, and he lowered the temperature of the incubator and showed that the thermogenic program was upregulated, and when he then raised the temperature, it was downregulated. And so what that proves is that these fat cells must have some intrinsic cold sensing mechanism. What exactly that mechanism is not clear, but there has also been really interesting work recently from Alex Pfeiffer and Bonn, which has highlighted sympathetic independent mechanisms for activating these cells either through adenosine or inosine. So I don't mean to imply that sympathetic nerves are the only way to activate these cells, but that's at least the most well-studied way.
Yeah, great. Quick other question. So for your genetic genetic variation study, are you focusing only on protein coding rare variation with the exome sequencing? Because as you know, we have worked in the common coding variation space where I guess Marcelo Nobrega's work and our work provides at least some evidence that non-coding genetic variation modulates beiging capacity of adipocytes. Is that of interest at all?
Yeah, absolutely. I met with Jose after I met with you, and he asked me
I know.
the same. And so We decided we had to start somewhere, and because we have more subjects with exome data than whole genome sequencing data, we thought we would start there. We also have perhaps some genotyping data, so we could have some incomplete
Okay,
whole
great.
genome data. But to start, we decided to focus on the exomes with the hope that we'll find rare alleles with large effect size, but we certainly don't intend to neglect looking at non-coding variants as well. And we value your expertise when we get to that.
Thank you.
Yeah, great talk. I had two questions, one specific, one a little more general. The first one is, do you predict that if you were to express PRDM16 in, say, like the lumbar region, will you shift the innervation pattern to innervate that region more?
Yeah, so great question. I think we would predict that if it's expressed in this crucial developmental window, perhaps yes. But we don't know for sure. And the reason we don't know for sure is because in these imaging studies I showed you, we haven't been able to do staining for PRDM16. And so what we'd really like to see is PRDM16 enriched in this highly innervated region or not? We think it may be, but we'd like to be able to prove that. And undoubtedly there are going to be other molecules it interacts with that are important as well. So I don't know if the one protein alone would be sufficient.
Thanks. And my second question is, you showed that there's a lot of genetic variability with how much brown fat we have at baseline. Do you anticipate that there will be similar variability in how much fat is inducible within people, or is there sort of like a ceiling at which everybody would hit?
Yeah, so great question. I think we don't really know. I'd like to think that both are under genetic control, but the quantitative data is harder to assess in large cohorts because in the clinical setting, we benefited from the fact that when the scans were read, the reader said brown fat was detected or not. And so we could do that binary categorization. If we want to actually quantify brown fat volume or activity, someone has to manually review the scan, which takes about 20 minutes. And so to do that for thousands of scans is an enormous undertaking. I'm hoping that it will soon be possible with automated or semi-automated approaches to do that, but we're not there yet.
3 more questions.
Okay.
So just curious, going back to the first part of your talk, since you had such a large cohort, did you analyze any factors such as exercise that may be associated with the brown fat distribution?
Yeah, great question. So because we had a retrospective cohort, we could only use the information we had available in the charts. We focused on diagnosis codes because those are readily available, and as any physician knows, because they're the bane of your existence, you have to do these after each visit. So we don't have clear granular data on physical exercise, but we would like to look at that. There was a paper recently published that just came out, though, showing that an exercise paradigm in humans was not, this was a prospective study, was not associated with the activation of brown fat. So it could be that that's one area where the rodent models and the humans differ.
Then is there any suggestion that brown fat protects against degenerative diseases?
Was it neurodegenerative or unclear? Yeah. So we are not working on that, but there may well be. There's a colleague at Augusta University, Alexis Stranahan, who's published some really interesting work on links between thermogenic fat and Alzheimer's in mouse models. And she shows that there can be changes in cognitive performance that presumably are due to some fat brain crosstalk that her group's working on characterizing.
Final question. Great talk. What do you think about the relative importance of thermogenic contributions from skeletal muscle versus brown adipose tissue?
Yeah, so I guess the point is that when one is cold, the normal response is to shiver, and that also generates heat. That's why we do it. And so if we're talking about heat generation, I think that undoubtedly shivering contributes more to heat. Our mass of skeletal muscle is way, way higher than our mass of brown fat. And so our prediction is that the benefits we see associated with brown fat are more likely to be mediated by signals to other tissues than by its energetic contribution per se.
Fantastic. There are more questions, but I think we have to end here. This
Okay.
was great. This was actually at the peak of it, I think 120 or so participants. So we had great participation between present and online. Paul, I can't thank you enough for a
Thank you.
fantastic seminar, and thank you all for coming. See you next time.